This morning we're going to do a plasmid prep. I've conveniently placed the procedure here on the in the lab. It's 225 Penske, and what we have here is we have cultures that have been growing overnight, growing about 16 hours, actually going on 17 since it is 5 a.m. at the moment. And we're going to do a duplicate of, say, 4 these cultures here, and place these. So these are the very first thing to do is we're just going to be simply pelleting, spinning these down. These are autoclaved 5 mil Argos epitubes. They were autoclaved again, so it's not so much a matter of sterility. They shouldn't have had anything in them previously. But we re-autoclave them just for extra measure. And so today I'll be working with basically a replication. It makes it a little different than if these were separate. We'd have to be really worried about trying to not cross-contaminate them. And so we can really just keep track of these as essentially A and B. So first, it'd be better if one of them was actually A. And first step is to spin them down, and that can be accomplished. These were actually placed in culture by simply touching a colony and then ejecting the pipette tip into the culture. And I'm just going to go ahead and pour that in. And grab the second replicate here. Problem there. Head, and these will be spun down for roughly 7 to 10 minutes. We have a centrifuge here specifically for this. This is referred to as the Argos centrifuge. This tends to vibrate. And I've found that little trick makes it not vibrate so much. So that's going to be going for roughly 7-8 minutes at full speed. At the same time, now is a good time to go ahead, and we're going to want to have elution water eventually for the plasmid, which we will have heated to 60 degrees. Since this was set up for heat shock, I'm going to go ahead and set it up for elution, and have to hit enter in order to get that to warm up. And this is depth sea water, so it's the water that has had the proteins deactivated by the addition of that chemical compound and autoclaved to get rid of it. And I've already pre-labeled these. We're going to have basically a series of lysis and precipitation There are two of the epitubes will be actually used for spinning away the protein and so forth, and the final tubes here have been set up for the eventual DNA plasmid after it has been eluted. This specific plasmid is actually a 10,000 base pair plasmid. So I'll come back on when we're done spinning. There's a series of solutions that we'll be using that are set up. These are actually aliquots that are normally kept actually up on the shelf up here. And so the buffers are referred to in various things. There's only one that's refrigerated. That's the P1, which is the initial resuspension buffer. And so that one I tend to place on ice. The others, which is lysis buffers, P2, and as you'll be seeing, we'll have some additional buffers for eventual elution, as well as neutralizing the solution, and these will be kept here for use as we start to work with the actual pellet. The resuspension step after this is the addition. Well, first of all, you need to basically pour off the bacterial waste, and so I'm going to keep a bottle right here for that, which we'll be adding bleach to. And you can see the size of the pellet that we're working with here, and it's trying to get rid of as much of the media as possible, because the media can give rise to interference actually. And so to do that, I'm just going to use a 200 microliter pipette, pull off any residual. Taking a look. There we go. And this is going to be resuspended, and the buffer P1. So that's the refrigerated buffer. So you'll see here it's 250 microliters and a P1000. And the process of resuspension. Actually, I'm noticing that media drop there, which I don't particularly like. So I'm going to go ahead and I'm going to add that for a second, and go ahead and remove that extra droplet. As far as resuspending, the goal here is to do this without necessarily gunking it on the pipette tip. So I'm going to pump up and down here, where I'm not really going to be stirring my pipette tip into that pellet, and it'll start to try not to aspirate too much air, but it will. May take a little bit longer if you don't work directly on top of the pellet. you can see because of the way we're aspirating this, this is a really good situation for, or a bad situation I suppose, for contaminating the pipettor. And so what I like to do is keep handy a Kim wipe, where there's enough on there to keep these things from cross-contaminating. So that's a resuspension of the first, and I'm going to go ahead and do the same now with the other pellet. And removing any residual. Go ahead and get that, as well as any that's in here. The way I like to think of it is a little media left may mess things up if I accidentally take a little bit of the cells. It's not a problem. Good there. Again, 250 microliters of P1. And resuspend that. The next step of this is going to be to actually lyse the cells, and the lysis phase is conveniently done with P2, and it's also 250 microliters. Oops. And actually I want to wipe that off. And since these are both replicates, I can do this certainly without concern for cross-contaminating. So P2 solution is here. So this will lyse the cells. Double check. And what happens next is that this is just sloshed, which various people's interpretation of that. I like to just slosh them back and forth like this, and that's going to be for 4 minutes. So I'm going to go ahead and say, okay. Okay, Google, set timer 4 minutes.
Okay, 4 minutes. Starting.
Okay, so I'll be doing this for 4 minutes, and then we'll come back to the neutralization step. Okay, so at this point we are going to go ahead and neutralize the solutions. For neutralization, there's the addition will be 350 microliters of the neutralization buffer, and that is N3. So make sure you unlock the pipetter, take it on up to 350, and this will be added to both. This is also an opportunity to contaminate your aliquot. That's why we keep these in small aliquots. And so if you contaminate your own, that's your problem. My problem, depending on how you look at it. That's N3. This will then be similarly sloshed for 2 minutes, and so okay, Google, set timer.
Sure. For how long?
2 minutes.
Okay, 2 minutes. Starting.
You'll notice the solution tends to get a lot of precipitate at this stage. And I'll do this 2 minutes. Okay, so we just got done the shaking of this for 2 minutes, and the next phase of this will be spinning out the precipitate. One of my students describes this as looking like more like shredded coconut, and that's done simply by pouring it into a EpiTube. These were autoclaved, again primarily to get rid of DNase, and at this stage you just simply pour that in. What I like to do is actually then take it and actually give it a little bit of a shake, and usually I can bring out an extra one, but it doesn't look like I will. Okay, so that's A, and you'll notice I'd marked these for the different stages to keep track of who's who. Got another drip there. These will be spun at high speed for 10 minutes. So I'll do that here. And tabs up. Don't forget the lid. And this will spin for 10 minutes. And we'll be back in a minute. Well, 10. So the next phase here just spun for 10 minutes. So now we will have pelleted out, as you can see, a lot of the cell debris and protein. And next, there's usually some floaters in this, and so typically run this through a second spin. And to do that, I'm just going to pipette it off quickly into a second tube. Notice the new centrifuge doesn't seem to make this as necessary as it used to, but yeah, there's some debris there. Just simply moving it over into the other tube. Since we're doing 2 spins, you can and don't have to be as conservative about pulling over a little bit of garbage. One and second one. And that's going to go back into the centrifuge for another 10-minute spin. And then we'll get to the actual column absorption. So now, after that 10-minute spin, the next thing is going to be actually absorbing the DNA onto a spin column. Now, I try to encourage in any work with DNA to try to keep your grubby hands out of the pot if you can. And so, in this case, having a couple here is helpful. Sealed up. Using the cheap knockoffs. That's good enough. I'll need 2 of these. So we'll be transferring the contents of that second spin to, in this case, there's just barely a small pellet that you may be able to see inside there. Back of my hand. It's nice to get that out though. There's a fan that stays on, so you should turn these off. This will be transferred now to the actual spin columns. We're going to keep track of A and B at this point. So at this point you do want to be pretty careful to not take any of the pellet over onto the spin column itself. So it goes on to the spin column. Plenty to wet it in this case. This is now going to be spun very briefly, 30 seconds, through the column, and then the plasmid will then absorb onto the resin. So this is just a 30-second spin, and for the 30-second spin, I tend to, one can just basically simply hold it. Don't forget the top. It's not good. And so rather than use the timer, I'm just going to actually count to 30. So after the 30-second spin, this is actually now DNA waste. This is not containing bacteria, and so we keep that separate. And so that represents what we've caught. And the next step will be to rinse it. And the rinse will be with 700 microliters of the PE buffer. I guess that would be pre-elution, maybe one way to think of that. So, and will be added to both of these. And then this will be done twice. To wash through the column. So it goes back in for another 30-second spin. And do the same. So there we have first rinse completed, and we'll get a second rinse of the PE buffer. And this will go back through for another 30-second spin. Should be opposite each other for balance. And off we go again. So here we're going to remove the second rinse, and it's going to be going for a final high-speed run to quote-unquote dry the column. So put that in there. And this is going to be a 2-minute spin. So this is a 2-minute spin. I'll use the actual timer. And then we'll come to the point of elution. So at this point the water is up to temperature, so we're good to go for the next step. So at this point the columns have been air dried, and you'll notice a very small amount of liquid that has kicked out of the bottom there. And at this stage the columns are going to be moved over from where they are now into the final tube. So be careful not to touch anything on there. And we'll do that with so-called depth seawater. This is A. This is B. That's been over here into the bath, actually. So we'll go ahead and grab that. And the amount of water that they recommend for elution is anywhere between 30 and 50. I like to use 31. Primarily because that'll give me a microliter to do the nanodrop to measure, and at the same time allow me to then have 30 microliters left over for use. Oops, 31. I like to go with a smaller volume because it will be a little more concentrated. It may leave a little of the DNA behind, but I still like it that way. There's a 2-minute incubation time after the addition. Now here the water should be added very carefully to the center of the column. We got the same plasmid in both, so we're not going to have to worry about that. So if you can see down inside there, but I'm literally going to try to drop it right down on the column matrix, and a 2-minute timer. So in 2 minutes we're going to spin that down.